Forests have been studied pretty well. We're able to accurately measure above ground biomass using remote sensing data or sort of ground truth manual measurement. But soil carbon is basically a new, it's a fairly new science to actually get rigorous and accurately measured um, whatever estimates over time. And as probably most of you are familiar with, like the carbon market has matured a lot. So going from sort of red plus projects to different levels of quality of carbon credits, we're now in a phase where the world recognizes we need to sequester a lot of carbon. Plants are a really great way of doing that, and that there are really significant storage solutions, whether they're in soil, biomass, the built environment, direct air capture technologies, and so we need more and more to be able to accurately understand how soil carbon works. So this is basically an overview of sort of existing literature, what we know now, and then maybe where the industry is heading, so that individual farmers, landowners can get paid for for increasing the carbon storage of their land as one motivation. So Colin, if you want to hop in, I would say go for it.
Sure, thanks, Troy. Is it all right if I share my screen so we can do just a
Yeah,
couple slides? Cool. Is that coming up?
that looks great.
Awesome. All right, cool. Well, thanks everybody so much for your time and your attention. I'm going to tell you a little bit about the work me and my team do, trying to understand how particularly which sort of fungi live in soils can affect how entire forests grow and the ability of forests to sequester carbon and lock it up in plant biomass, and especially lock it up in soils. And to begin, I want to place you sort of in a forest, and I just want to emphasize that most things we know about forests are based upon things we can measure above ground. So for at least 100 years, ecologists would walk into a place like this, and they might count the number of stems in this forest, identify the species, they might measure how big they are, and now we'd probably remotely sense features of this forest canopy from space. And all of this totally makes sense. Above ground in a forest is where photosynthesis happens. And this is the primary channel by which, carbon enters terrestrial ecosystems. Plants are converting carbon dioxide into photosynthetic sugars that they use to build biomass and grow. But if you only ate carbohydrates, you would be a very, very sick human, and so would a tree. Trees need minerals and nutrients, particularly nitrogen and phosphorus. And in general, they can't get those above ground. They have to get those below ground from the soil. And to do that, trees build roots. And trees and all plants build a lot of roots. So in many ecosystems, there can be as much or more plant biomass below ground in root structures as above ground in stems and leaves. So this lower picture is a forester using a tool called an air spade, blowing away just this top layer of soil. And what he quickly reveals is this massive and this overlapping network of roots. And it becomes super clear pretty quickly that there's this huge potential for really important ecological interactions to be going on below ground, as well as really important carbon dynamics. However, when you look closely at these roots, and I mean very closely, like you'll need a microscope closely, you realize that they're not really just roots at all. They're fungi. So most plants on Earth are in symbiosis with these symbiotic mycorrhizal fungi that grow on the roots. And what's going on here is they're trading. They're trading some of those photosynthetic sugars in exchange for nutrients these fungi are exceptionally good at acquiring from soil. And this is just part of the picture. Lots of trees, especially in the tropics, also partner with what we call nitrogen fixing bacteria, which actually live in the little circular nodules in that image. And that allows trees to access dinitrogen gas that's just in the atmosphere, but it's really hard to break down. But basically, these bacteria can convert it into plant usable forms. And these symbiotic microbial partners of plants and trees are embedded within an even more complex free-living soil microbial community that also includes soil animals like nematodes, but also fungi, bacteria, archaea, viruses. And soils are actually the most microbially biodiverse habitats on the planet. It's truly incredible. And in totality, this is the forest microbiome. And we're coming to discover, just like we've recently discovered in human health, that which microbes live in your stomach can dramatically affect whether you're sick or not, whether you're healthy. This is also true in forests. Which microbes are in soil, we're coming to discover, can really affect the capacity of forests to sequester carbon and lock it away in tree trunks and lock it away in soils. And I'm going to dive just a little bit deeper into some of the research we do on that. But first, I just want to remind you a little bit about and put this in the context of sort of the global terrestrial carbon cycle. And so annually, we emit, by burning fossil fuels and land use change, converting natural ecosystems, usually to agriculture, about 9 gigatons of carbon dioxide to the atmosphere. And of course, this is the greenhouse gas that's primarily responsible for climate change. And so that's a lot of carbon. However, it's important to realize also that there are natural fluxes of carbon dioxide on Earth, and they are huge. So every year, through photosynthesis, globally, all the plants on Earth sequester about 63 gigatons of carbon and convert it. Basically, when that happens, they're scrubbing it out of the atmosphere so it can no longer contribute to climate change because it's now in a new form that's building plant biomass. That number's huge, right? Like that's almost an order of magnitude larger than human emissions. But again, the terrestrial carbon cycle is a cycle. Trees don't live forever. Eventually they die. And when trees die, they enter soil carbon pathways. And then those free living microbes we were just talking about eat dead stuff. They respire CO2 back to the atmosphere. And that is also a massive flux of carbon dioxide. And so on the net, terrestrial ecosystems actually sequester around 3 gigatons of CO2. So they basically, all the plants on the earth, and especially forests, do us a huge favor and offset about a third of our global emissions. And we're coming to understand that the soil part of this whole picture is really important. So when we think about where carbon gets stored in restoration, we often think about trees. We see a tree growing and it makes total sense, but actually so much of the carbon on Earth that's sensitive and that's important for natural climate solutions is in soil. This is a figure that we put together for a paper we recently did on the importance of soil microbiology for understanding soil carbon. And to try and illustrate this, what we did is we took data on the global distribution of plant biomass carbon, and we just binned it up and summed it up by latitude. So basically the left side of this figure is the South Pole, the right side is the North Pole. And what you see is there's a peak in plant biomass right around 0 degrees, around the equator. This is where the Amazon rainforest is, and the dipterocarp rainforest of Southeast Asia and Central Africa, right? Huge amounts of carbon density there. Then we did the same exact thing, but we did it for the first 2 meters, so about 6 feet of soil. And this is what that looks like. There's just way more carbon in the first 2 meters of soil than all of the vegetation on Earth, as well as the atmosphere combined. And you see, yeah, there's a lot of carbon in the soil in the tropics, but also you see this huge amount of carbon in northern latitudes. What that is boreal forests that build these huge organic horizons, as well as Arctic tundra, where you start having permafrost dynamics become really important. When soils freeze, that basically stops decomposition. And so there's just this massive amount of carbon in the soil. And this is the part of the terrestrial carbon cycle we understand the least. And it's because it's the hardest to see. It's literally, you have to dig it up. It's below ground. We're also coming to understand, though, that a big part of understanding why carbon is in some places and more abundant in some places than others in the soil has to do with which microorganisms are there. So this is work I did with collaborators back in 2014, where we figured out that particularly which fungi are in soil can dramatically affect how much carbon is locked up in that soil. So if you had 2 forests that had the same climate and the same soil parent material and very similar species, but one was forming, symbioses with one type of mycorrhiza versus another type of mycorrhiza, that can result in 50 to 70% more carbon being locked away in that soil, which is absolutely huge. We're also coming to learn that these microorganisms affect how much carbon gets locked above ground and the ability of just the input pathway as well through the trees. So this is work I'm doing with the Crowther lab right now in Europe. All these points across Europe are forest monitoring sites run by foresters, and this is a picture of one actually, where basically foresters go out and have since the 1990s been measuring tree growth. There's about 300 of these sites that we're working with. And we've convinced them all to go sample soil these past 2 summers and mail it to us so we can sequence DNA in it and figure out which mycorrhizal fungi are there. Because we want to know, does it matter which mycorrhizal fungi are there for how quickly these trees grow? We actually thought maybe certain mycorrhizal fungi invest more in nutrient acquisition. They're just better, quote unquote, symbionts. And maybe that would make trees grow faster. And so what we did is we fit statistical models like most scientific analysis like this would do of tree growth to things like climate, so how warm or wet these places are, soil nutrient pools and tree size, and all this other stuff that we know is important for tree growth. And then we started using that molecular DNA data to sort of identify really important features of these fungal microbiomes. And here we're plotting tree growth rate. So these big circles are just the average tree growth rate of the entire plot. And then we have actually the individual trees, some of the individual trees within those plots, just to give you a sense that this stuff is incredibly noisy and that's normal. And you start to see these signals when you scale up, when you start thinking about hundreds and thousands of trees. And we find that, so on the x-axis here, we have this ratio of nitrogen to carbon metabolism genes in the mycorrhizal community. You don't actually need to know what that means. What you do need to know is that basically, if you're on the right side of the graph, you're partnered with certain fungi that are linked to you growing a lot faster than you're on the left side of this graph. And that's a log scale. So the outcome of this is actually a doubling of carbon capture rates, depending on which fungi are on your roots, which is crazy and has led us to start thinking about well, man, maybe we need to start moving these things around. Maybe we can start engineering forests by manipulating their microbiomes to sequester carbon faster in stems as well as in soils. And it's also pushed us to start thinking about some of the restoration work we do and start thinking about not just restoring the trees, but also restoring the forest microbiome. So when we go to a tree planting project and we're restoring a forest, we go there and we have seedlings and we drop trees in the ground, but we never plant, quote unquote, the microbiome. But again, we know these organisms are critical to how those trees access soil resources that are often critically limiting. We know many trees can't establish in the wild at all without their symbiotic partners. And finally, most places we're looking to restore look something like this. So this is actually one of our experimental sites in Wales, prepped for planting in the UK. And it used to be a sheep grazing pasture, and it's been that way for at least a century and probably many centuries. Most restoration on Earth is happening on ex-agricultural land, so land that was used to farm or graze animals generally. We know the microbes in these soils look nothing like the microbial communities of the intact forests we're trying to restore. And so that sort of begged the question for us, what if we changed that? What if we introduced those microbes? And so we've started doing randomized controlled trials here as well as in Mexico. And we're trying to set up a few more sites right now to test this really simple idea. And so what we're doing is we're doing tree planting business as usual as it's done in the UK right now. Or we're doing that tree planting and then we're adding a small scoop of soil that we've sourced from a nearby ancient woodland. And the point is to reintroduce those mycorrhizas and those other free-living microbial community members into that soil. And our question is, will this increase the probability that the trees we put in the ground stay alive until next year and the year after that? Will they grow faster? And also, we're really interested, we actually think this is going to massively affect how much carbon gets sequestered in the soil for a lot of reasons. We think it's basically going to happen a lot faster because we know these fungi can connect soil particles. There's a lot of mechanistic reasons. But to get that number, before any of the site was prepped, we've already taken hundreds of soil cores across this site to sort of get that baseline soil carbon information. And doing that took specialized equipment. It took people really knowing what they're doing from the scientific community, going out there in person to make sure it was done right. And to document potential soil carbon changes, we're going to have to do all of that again in 3 years, and 5 years, and 10 years. And so we're going to answer this question, but we're going to answer it really slowly, compared to the timescales we want to put carbon back in the earth. And it's gonna take a lot of people hours. People have to manage where all those soil samples are. They have to keep track of them. They have to actually send them out to laboratories and prepare those samples. That costs real money. And it's just a hard thing to do. And it brings me sort of this question Troy was asking me the other day is, how do you count soil carbon gains in these restoration projects? Hopefully you don't have to do what we're doing there. We're doing that because, it's a scientific project and we've put so much into it that it's really valuable for us to get that number very, very right. But even if you surmounted some of these problems and made it a bit logistically easier, there's still a lot of stuff that needs to happen and a lot of chemical analysis to do that right, and it still happens slowly. And so it's totally a bummer because there's so many restoration projects out there who are reporting the carbon they think they're sequestering above ground in the trees. And they're certainly most likely building carbon in their soils, but it's just beyond their means to quantify it. But if you could potentially have a huge impact on how much carbon they're able to report they're sequestering. And we know that because our group has done analyses on how much carbon globally you can sequester. If you took all the area so this is basically a map of the earth where if you put trees back everywhere we think you could reasonably put them back, this is what forest cover would look like. And we think you could add about almost 1 trillion trees, and that would sequester 205 gigatons of carbon. So that's about 1/3 of all the carbon we put in the atmosphere to date, which is a ton. But when people hear that number, they're like, sick, we'll plant some trees and the carbon will go into the tree. And what they don't realize is when we calculated that number, we considered the whole ecosystem, including the soils. 51% of that 205 gigatons, so a little over half, is actually soil carbon. And so if we're not counting it, and we're not actually valuing it in these projects, we're missing potentially half the value at a global scale for that carbon. And so when I get asked this question all the time by restoration projects, how do we do this? How can I value this at my project? And my answer is, I think if this is going to be done right, the answer is you don't. You don't try and count soil carbon in an individual project because it's so hard. Instead, what I think you do is you build and you value a portfolio of projects and you answer this question probabilistically. And I'm going to explain what I mean by that. So this is data on how much carbon might be able to be restored if we restore places to natural vegetation based on analysis by Jonathan Sanderman, basically in metric tons of carbon per hectare. And this is the distribution. So if you just look at the global surface where you could restore soil carbon, most places, if you restore natural vegetation, you're going to get a carbon gain. Some places more than others. Some places you'll actually lose it. But on average, in total, it's a big gain. Now, we know some stuff about which ones are probably going to build it, probably going to lose it. But let's say you didn't. Let's say this is all you knew, this picture. You could still put together a portfolio of projects where you could become reasonably confident that you're going to build soil carbon. You just need 100 or 1,000 or maybe 10,000 hectares or something like that. But then you will know with some level of certainty and on some uncertainty range, and you can collapse that based on how many you jam into this portfolio, you can become very, very confident in the number. And this is sort of how people monetize securities that are based on mortgages. Trying to predict whether an individual person is going to default in their mortgage is analogous to trying to, predict a coin flip. It's something you can't really do. But you can put together 1,000 or 10,000 mortgages and say, oh, the default rate's like 2 or 3%. So now I know how to finance this, and now I can make this a product. Essentially, you take the same approach with soil carbon. And what that looks like in practice is, some projects are going to be punching way above their weight, sequestering a lot more carbon than maybe they're getting value for. And some projects will be getting value for carbon, even though they're actually causing carbon losses from the soil. But the point is, in the totality of all of it, you're getting the answer right. And these things, the accuracy goes up with scale. And so the value here is that neither of those projects was getting any value out of that carbon before because they had no reasonable way to be confident they were building it. And so I think this is one really cool way to start thinking about this. Many of these problems with carbon and ecology are fundamentally problems of scale. Things in ecology in general are known to just be impossibly unpredictable at the scale, at very small scales. I can't tell you what species an individual stem in a forest is, but I can tell you the frequency of that species across, 1,000 hectares. And so I think we need to take these sorts of scaling approaches to really getting these values right and actually accurately representing how much carbon is there. So with that, I want to thank you for taking the time and listening to me talk about fungi and soil carbon, and I'd be happy to answer any questions about this stuff.
Awesome. Thanks so much, Colin. Maybe just like a quick recap for me. I was impressed by the magnitude, both the magnitude of the actual soil carbon in the ground, but also the magnitude of the change based on different fungal types. And so I was just like, okay, that's a huge number, and I've known it, but it's like having a visceral understanding of it is a bit different. So I don't think Elaine's on the call, so we're just going to jump right into questions. So if people have questions for Colin, just unmute yourself or write it in the chat, and we'll just start taking questions. So one question from Liron is: do you believe that seeding existing forests or reforestation projects with a better microbiome would create additionality in projects that otherwise wouldn't qualify? Maybe you can just talk about that.
Yeah, I think potentially yes. So the reason we're doing these experiments is because we think it really might matter a lot, for a lot of reasons. We know there's some work out there showing that there's some places that are exceptionally difficult to restore, and people have shown in heathland communities in the northern UK, as well as trying to restore grasslands in like really degraded ex-mining soils in eastern Estonia, that they just can't get plants to establish there and restore those communities until they move some soil inocula. And then they actually get successful restoration. And the thing we're bringing to these projects is both the restoration angle, but also the carbon angle. We think the reason those things are happening is because they're allowing these trees to establish better. We know that they're going to be building more complex soil microbial communities, which generally increases soil aggregation. There's all these mechanistic reasons why carbon sequestration should go up, but these things just haven't been tested very much. So I think it would be premature to say that is true, but I think it probably is.
Cool. I know Matt has a question. Why don't you chat?
Yeah, thanks. So I think this is a really great idea. I have definitely not thought of it this way, and I think this could definitely make things easier. I guess I have two questions. One of them is, basically you're relying on this kind of baseline of data, and I could see a scenario where people get much better at it. They use your research, they understand like, oh, we need to plant the fungi as well as the trees. They get much better at storing carbon underground. That kind of baseline or aggregate of projects doesn't really take that into account. So you're not really taking advantage of the improvements and understanding with drawing down carbon. So I guess like one question is kind of how do you address that, or do you think that would kind of just naturally move in the average over time?
So you mean you're worried that even if they did this, they might not be able to realize the carbon gains or something? Or
no, so in the distribution of projects that you have that you're basically going off of to say, okay, every project is averaging whatever amount of carbon drawdown. As you get better, right, that distribution will change. So how do you take into that account, or do you just kind of write that off, or do you just keep on, like that? The baseline is all these projects that you're aggregating, and the best, the statistics around that are going to be much different as you improve the process over time.
Yeah, so maybe there's some bonus we're creating in soil carbon sequestration when we do some of these new things we're trying, like microbial inoculation. And if we take the approach I advocated at the end, we're going to miss that bump if we don't actually measure it. And I think that's absolutely true. But I think there's other reasons to do it. And I think if we miss that, like if we end up having a conservative estimate of the carbon, I think that's okay. And there's actually more. I mean, that's just a win. And I think the other thing to remember in all of this too is that we're not just, I mean, it's awesome that restoration creates this awesome carbon outcome, but that's not the only reason we're trying to do it, right? Like we're facing two global crises. Like one is the climate crisis. The other is the sixth mass extinction event, like of life on Earth. We want to restore nature, and if these things lead to the increased survival of these trees in the field, that's a win. Also, when we talk about restoration and restoring these biodiverse communities, I think when we say that, we don't just want to restore the tree composition. We want to restore the whole ecosystem composition, and that includes which insects are there, and which macro animals are there, like jaguars or something, and that also includes which fungi are there. And so I think for all these reasons, like, yeah, maybe we'll miss it in the carbon valuation, but I think if it works, I think there's many more reasons to do it beyond that, just that carbon accounting.
I love that point, Colin. It's easy to remember in like the climate tech community that we're not solving for carbon. It's just that carbon's what we can measure and get paid for. But the actual fundamental issue is a degradation of nature and imbalance. So even saying that, we do have a couple questions about carbon measurement. Basically, um, are there going to be cheap and accurate ways to measure carbon, or do you know of any technologies where we don't just take a large statistical survey based on like small training sets, but actually physically measure carbon in individual projects, and what you think of the future of that?
Yeah, so I think right now, I mean, the gold standard for measuring carbon in a soil sample is you take a soil core, um, you process it, you dry it out, and then you basically combust it. And that in a very high temperature oven, and you measure the CO2 gas that comes off. All the carbon gets converted into CO2, and then some analyzer can detect that. And that costs some money. But there's new ways people are doing this. Basically, a soil that has more carbon in it often looks darker. And so basically, there are spectral properties of the soil. So people have done cool stuff with near infrared spectrometry. There's basically these cheap little scanners that rapidly you can zap a soil in just a second with very little prep, and it gives you a number. And then if you have a training set of soil carbon for that site, you can build a machine learning model and actually get very good accuracy. And so if you're going to be taking a ton of samples at a site, that works. But the problem is it doesn't generalize to new sites. Like every soil type, every place needs its own calibration. So it doesn't, solve this in the way like, blowing up the sample always works. The other problem you're having is soil sampling is fundamentally a destructive process. It's like if I wanted to measure how much you weigh, I had to totally incinerate your body. I could never measure you again. And so I can only measure, whether you're gaining or losing weight by measuring a population of you. And the problem is there's just a lot of variance. From here to here, you might go from 2 to 4% carbon in the field, which is just normal heterogeneity because who knows why, right? Like there's some more rocks over there than there is over there or something like that, stuff that's just never going to be predictable. But if that whole field gained 2% carbon, that would be a massive carbon gain. And so there's just some of this heterogeneity problem is insurmountable because of the destructive nature of sampling. So like if you invented something where you could like just Star Trek scan the soil without ever disturbing it and somehow measure carbon, like that would be a game changer. But I don't think that tech exists, but someone should make it. But yeah, so until we do that, we're basically relying on stuff. I mean, the other thing to remember though, it's not, the training sets aren't a couple hundred samples. There are hundreds of thousands of soil carbon samples from all over the world with great coverage in digital repositories on the internet. And you can build pretty good machine learning models to those. And so you can, the challenge is figuring out the temporal dynamics and figuring it out at an individual project. And it's just still hard.
Yeah, maybe I can just chime in there. So just providing also a bit of context for this call, like we invited Colin to give an overview and also be a sounding board for the Earthshot Labs project, which is essentially doing exactly what you just said, taking existing literature, data, and the hundreds of thousands of soil samples from around the world, and build a carbon sequestration map, just like we've done with forests, and try to actually have the world and individual landowners and policymakers and investors understand the potential for soil carbon on their individual piece of land. And while it's very expensive to then go say, okay, we're going to measure every single piece of land on Earth, across the globe, we can be quite accurate, at least along the bell curve. And so that's sort of the first sort of MVP that we'll be working on this week. There are also a couple questions just basically about implementation. Like, okay, so say you've got this, sheep farm or, pasture or rangeland or degraded soils, mines, etc. What does the actual implementation plan look like for restoring, mycorrhizal fungi or soil microbiology? And like, what are some different techniques and how expensive or effective are they?
Yeah, I mean, we're super like early in this. We're just scientists, so we're never thinking about like, oh, how do we scale this financially? That never crosses our mind. But yeah, right now we're moving soil because it's straightforward, right? Like we're moving a whole community. It's very practical. We're not adding a lot of soil, so it's not like there's some mass excavation somewhere. But other people have established, you know, particular subsets of fungi in the field before. Um, so like actually truffle orchards do this. Like people love truffles and they're worth a lot of money. And those are a mycorrhizal fungus. That's one of the reasons they're so expensive. You can't just farm them somewhere. They need to be in symbiosis with a tree. And so people do successfully establish truffle orchards. And that's an example of people manipulating the below ground microbiome and changing it to be something they want. There's also some examples in forestry in France where people have done this back in the 90s and early 2000s. They established forest plantations with of the same species with very different mycorrhizal communities, actually single species, and they're able to show that even 20, 25 years later, those same fungi are there using molecular stuff. But there are people selling microbial products too, especially in agriculture, a ton. This is easier with bacteria to generate mass cultures. The tricky thing is there are some products I've seen that are rigorous and do have good scientific testing and seem to be totally legit. But I would say for every legit one, there seems to be like 20 like half-baked weird ones that just like, I'm like, maybe this is doing something, maybe it's not. It's not clear. It's more challenging for fungi, but it's not insurmountable. And like, that's a big question I have right now. Like, how do we scale this, especially for the work we're doing in Europe? Like let's say we find indicator species that are really good and actually on a signals of causing, changes in tree growth. Yeah, how do we scale that? And that's a great question. If you know someone who can answer that question, I want to talk to them.
Cool. Yeah, we also had another question about agricultural systems. So, in the soil carbon, market right now, there's a few big players that are promoting agricultural systems like cover cropping and no-till as ways to increase soil carbon, or at least not lose soil carbon. But are there other, agricultural interventions or methods that you think are worthy of starting to basically implement and aggregate for the carbon market? So, cover cropping and no-till is an obvious sort of like step, but then I wonder if you have more.
Yeah, so those are great solutions. It'd be awesome if they were more widely adopted because yeah, they do actually work. There's a ton of interest in this. Again, like, when we think about agriculture, like we've spent a century selecting on the corn genome essentially to the point where now like there's almost no variation left in most of our main, corn strains that we plan out, like so much that they have to introduce new genes because they've selected on it so much. But we barely touched any of this microbial and this microbiome side of it. So a colleague of mine at Texas A&M, Greg Sword, works with foliar endophytes. These are fungi that live in the leaves of everything. And he was doing work showing that if he could inoculate with particular fungal endophytes in these fields, he could, under drought conditions, so these things become really important by creating all these like metabolites that affect osmotic pressure in the leaves. So under drought, they can be really beneficial. He could get like a 10% increase in yield in corn. And no one believed him at first in his department because, if you get a 1% to 2% yield increase off of genetic manipulation, they're stoked. That's huge. So it's clear to me that in a lot of these systems, there's big levers that we haven't pulled yet, and they're the microbial levers, just because it's so new. As for soil carbon sequestration, I think it's anyone's game, but I would be shocked if there wasn't something on that end. So I know we were just meeting. Caltech is starting a new institute that's focused on soil, and this is what they want to do. They want to hire a ton of people to start doing microbiome science to figure out how you manipulate the carbon cycle in agriculture. This is happening. And then, people I used to did my PhD with got scooped up by Monsanto. They're doing this for them, mostly on the yield side, not on the soil carbon side. But this is definitely this is happening. The other thing, though, is it's a huge, first principle science question right now. Like, there's just a lot of research that needs to happen first.
And maybe that's a good intro. So, the Earthshot Labs community, there's basically a bunch of very smart technical people, software engineers, data scientists, investors, like a lot of people who want to give their time to addressing climate and ecological issues through technology or other means. And I wonder if you could sort of like identify, are there particular problems in academia or scientific literature or more like applied science and agriculture that are most worth working on?
Yeah, I mean, I think the biggest thing in this space that comes up all the time is that even with the philanthropy dollars that are out there, the, people are having a hard time finding enough good projects out there to give to. Um, we, Setting up these large scale restoration projects is hard, like for many, many different reasons. Part of those questions are ecological, like which tree species do we grow? How do we do this so we're actually restoring real biodiverse places? And those I think are surmountable, but there's these other ones like how do you go to a new place and like set up nurseries and recruit crews of people to do this and project management that team like in a way that actually scales and doesn't require you to like just spend 3 years messing up. How do you The other question is at some point, right? Like this is all well and good when we're talking about, a small set of projects, but if we actually start doing large scale restoration, like people are proposing, like there's this whole social side of this problem too, right? Like we shouldn't be buying up land and just saying this is only for this, and I took a bunch of money from the global north and just bought a bunch of land in the global south, and now a bunch of people who used to farm there like don't have it. Somebody needs value from those things, and there are these underlying social reasons why deforestation happens in the first place, right? Like there's a reason people clear cut and slash and burn the Amazon. It's because they're poor, and it's subsistence farming, and some of those things need to be figured out. Like how do we create models like, and I think this is what you're talking about, is like that give those people value for the carbon, for the biodiversity they're restoring, for water services, stuff like that. Are there other hybrid models you can do with agroforestry or other plantation stuff? How do you use the restoration to solve both this carbon and biodiversity problem, but also this social problem? And so, yeah, like I think we're going to figure out the ecological stuff. I think remarkably that's the surmountable part. I think it's the interactions with the scaling of it, both logistically on the ground when you're going to these places you don't know, and socially.
Cool. So we have a ton of questions that just popped up. We have an offer from John Headland of TriPoint Ag, who says he's got some interesting implementation mechanisms that he might want to share here. So you're welcome to unmute yourself and chime in, or we can take a couple more minutes. We have questions. What is the effect of biochar on soil? Do you see this as another one of these big levers? I'm also, I also deeply care about the social equity aspect. Like, so why are we destroying nature anyway? Um, you know, economic justice is racial justice, is environmental justice. Like they're all linked together. And I think that's one of the future of Earthshot Labs. Maybe we'll be doing direct project development ourselves or supporting other organizations, with technical expertise. But each of these conservation projects won't work in a multi-generational way unless the underlying issues are solved.